Thank you for the kind introduction, and also thank you to the organizers for the invitation. So the topic of my talk will be on sedimentary DNA, and I'm going to cover major past achievements in the field, and I'm also going to talk about some of the ongoing challenges and some of the future directions in the field. All right. So if at the end of today, when we all leave this room, if somebody came in and took swabs of all the surfaces here, let's say tables and benches, it would actually, we would stand a very good chance to get DNA from all of us here. But if somebody sealed this room and then comes back hundreds of thousands of years after, actually the chances of retrieving our DNA would be significantly reduced. The situation would be different, though, if instead of this carpet, we had some soil or some sediment. And it's going to become clear why that's the case as we go along. So sedimentary DNA is a concept. I mean, even if you're not familiar with it, from the name itself, it's obvious it's basically DNA that we retrieve from sediments. But to understand its novelty and significance, it's actually important to go back, and not so much back because it's a relatively young field, only about 20 years, but to go back to how the field was born. So the field actually was born out of two very novel ways of thinking about DNA, as sort of breakthroughs. So the first one was the idea that DNA can be retrieved not only from living organisms or recently deceased organisms, but also from fossilized remains of long dead organisms. And this idea came first, as far as we know, to Professor Alan Wilson from the University of California, Berkeley. And he basically tried to retrieve DNA from a skin of a zebra-like ungulate, quagga, and he succeeded in 1984. And since then, ancient DNA has been retrieved from a variety of fossilized remains, bones, teeth, hair. And so that was the first major breakthrough. And then specifically for the field of sedimentary DNA, about 20 years after, the idea from Professor Esquivireslev was that since we are shedding our DNA constantly into the environment, not only us, but all organisms, some of the DNA gets into the sediment. And so perhaps it gets preserved there and it is possible to retrieve it. And so that's what he tried to do and succeeded in 2003. He retrieved DNA from Holocene and Pleistocene sediments from Arctic and from New Zealand. And so that opened up really the field of sedimentary DNA. And since then, we have retrieved DNA as a community from a variety of sedimentary deposits, lake sediments, marine sediments, cave sediments, even speleothems and ice cores. And today, the discipline has grown a lot, especially over the last 10 years. We have seen a major uptick in the volume of ancient environmental DNA studies. Also in 2022, Ancient Environmental DNA has been a runner-up for the Breakthrough of the Year award done by Science Journal. We also now have Sedimentary DNA Society, and that regularly meets, has sedimentary DNA meetings. So basically over a course of not so long, 20 years, the field grew very fast. And so you may, well, before we kind of tackle why, the first question is why actually sediments are so good for capturing and retrieving DNA. So this question we are only starting to unravel, so there's a lot of unknowns, but preliminary data indicate that a key mechanism is basically adsorption of DNA fragments onto mineral particles in the soil or sediments. And so you can see here on this graph the absorbed DNA amounts at the equilibrium concentrations. And you can see that there's quite a bit of variation in the adsorption capacity among different minerals. So for example, smectite is one of the best ones with the highest absorption capacity for DNA. Chlorite, not so much. And of course, quartz, it's actually quite low. And also, this work has been done and has been led by Associate Professor Krina Sen, a colleague from University of Copenhagen, who is specializing in mineral DNA interactions. So she basically looked at this question, and she also visualized very nicely how DNA attaches to chlorite and other kinds of minerals. Okay, but why would we want to have DNA from sediments as opposed to DNA from tissue and other kind of fossilized remains? Well, there's 3 reasons that I will highlight here. The first one is that it's able to detect soft-bodied organisms, and as many of you know, many of the foundational or keystone species in ecosystems, they basically have soft bodies, so they don't leave fossilized remains, but they do leave their DNA. So a nice example is this study from 2024, also colleagues from Copenhagen. They looked at a core they collected from Greenland area. It spanned the last 11,000 years. And they wanted to see presence of an algae, a dinoflagellate, Polarella glacialis, because it's an important proxy for sea ice distribution. And so traditionally, they would be looking for cysts, but they only found cysts in the top and in the bottom of the core. But when they looked at the DNA, they actually found DNA of this species throughout the record. So this actually really showcases the strength of this method as a complementary proxy to others, because otherwise you would think that actually for most of the time at this particular site, there was no sea ice. But even for populations that actually produce a lot of fossilized remains in the fossil record, such as mammals, when their populations dwindle, that means that there's like just only a few last individuals in the landscape, the chances of detecting remains of those last few individuals is actually quite low. Again, because those even last few individuals share their DNA throughout their life, we actually have a chance to capture their DNA. And this was nicely shown in a study by Wang et al. In 2021. So we looked at the whole Arctic. We had about 500 samples from lake sediments and permafrost, and we wanted to see biotic communities and changes in biotic communities over the last 50,000 years. So we detected different plants and animals in the record, and one of them was mammoth. And so when we compared the sites, which basically show mammoth presence based on eDNA, which are triangles, with those that are based on fossilized records. So if we look at LGM and pre and post, we can see a lot of sites that basically either we have eDNA or fossilized remains. But as we go into the Holocene, not only did the number of sites drop significantly, but we also see some regions that actually we don't have presence of mammoth based on fossilized remains, but the ancient environmental DNA suggests they were there. So again, a nice case where DNA can complement other proxies. But perhaps the biggest advantage, and why maybe this proxy has become so popular over the last 10 years, is because it is possible to reconstruct an entire ecosystem from a single sample using a single method. So we can actually get DNA from bacteria, viruses, fungi, all the way to higher plants and animals. And so that allows us to build whole trophic levels and really look at, in more detail, but also in a larger scope, ecosystem dynamics. And just to give you an idea of what would be a typical workflow for ancient sedimentary DNA, so we would start with the sediment core, as we would with many other proxies. We would subsample it. Then we do directly DNA extraction from the sediments. We build genomic libraries, and then we sequence the DNA. After that, we do bioinformatic analysis. So we start with quality control. We remove any reads that are duplicates, that are too short to be taxonomically significant or unique. And then we move on to mapping. So basically what we do, we take the ancient reads and we map them on the reference genomes of different species, basically all that exist. We don't have reference genomes of all species, and that is a challenge, but I won't go too much into that now. This is the most computationally demanding step. And after that, we also have to authenticate DNA, so we have to check whether it has the damage patterns that we know ancient DNA should have. So it is not the most economic workflow from the expense point of view, but it is in terms of time and effort. And a good example of a whole ecosystem reconstruction from our section is this study from 2022 in Nature, Kier et al. So we retrieved DNA from a layer that was dated to 2.3 million years old in Greenland. And we reconstructed plant and animal assemblages at that site. So you can see here all the different plant species. I actually apologize if it's not very visible. Some of them were found already with pollen and with macrofossils, but many have not. One example is populus or poplar, which is a tree species, and so it would suggest that that area was more of a woodland than previously thought. But the biggest gain was actually through looking at animal communities, because there were very few fossilized remains of animals in this layer. So we found DNA of geese and reindeer and hare. The biggest surprise was actually that we found DNA of mastodon. And so we actually, based previous fossilized remains, we were not aware that this animal was up there in Greenland. So this is actually the most northern point of presence for this species. So this was quite exciting. So DNA has many advantages, but it also has some challenges that is worth mentioning. So, of course, the first challenge, just by being ancient DNA, it means that it's fragmented, it's damaged, there's less of it than for modern material. But even if we compare it to macrofossil DNA, so DNA we would get from fossilized remains, there is an additional challenge. So if we retrieve DNA from bones or teeth, we consider this sample a genomic sample, meaning that it belongs not only to a single species, the DNA, but also to a single individual. When we retrieve DNA from sediments, we are actually getting DNA of many different species, hundreds to hundreds of thousands of species, depending on the diversity of the ecosystem. And not only that we have many of these species, but we also have multiple individuals of those species that are contributing their DNA. And while we are very good at, or fairly good at actually assigning to reeds the species they belong to, so we can actually say these reeds belong to species 1, species 2, 3, etc. What we are not able to say at the moment, and it might not be possible ever, we'll see, is to actually know how many individuals contributed to, for example, species 1 pool. And we also cannot assign individual reeds to those then individuals. And as a result, we don't have single genomes. And that is a problem because most of the tools that we have and methods in population genetics are actually developed for single genome kind of data. So that means that actually we need to modify the existing tools and methods and maybe even build some completely new ones. So that's one option that's on the bioinformatics side. But there is another way, potentially, and that is that we know that we have tiny fossils in sediments, microfossils. And they come from a variety of organisms. We have single-cell organisms such as diatoms and ostracods and foraminifera. We also have reproductive units such as pollen and fungal spores. We also have pieces of larger organs such as leaves and seeds and shards of bones. And we could potentially get DNA from those single pieces, and then we would know that they belong to a single species and a single individual. But the potential of these kind of remains to generate single genomes is largely untapped. And the main reason is because it's technically very challenging to get DNA out of microfossils. Because if you remember from the previous slide on workflow, we would basically take sediment samples and then we would just extract DNA directly. But here we first have to isolate microfossils, we have to sort them out in their individual reaction chambers, we also then have to lyse them. And many of them are hard to lyse. They have a hard shell. That's why they get preserved in the fossil record. So we have these 3 steps, and neither of these is a trivial task. And only then we can go to DNA extraction. And so as a result, there's only one study to date that has tried to do this, and tried to do this on pollen. So it's a methodological proof of concept from Bennett and Parducci in 2006. And it basically consists from taking a pipette, extending it over a flame to get it like really thin. These days you could get the micropipette, but then you do everything manually. So you actually pick up individual pollen grains with that micropipette, put them in an individual chamber, like a PCR tube, and then under the microscope you have to find it again, and you have to break it open with that same micropipette. And you can imagine human doing that, grain after grain. So it's very laborious. And also then in the end it's very disappointing, because the follow-up study showed, so they actually looked at, I believe, over 1,000 grains, or close to 1,000 grains, and only a dozen worked. So it's a success rate of 1.5%, about that. So that's quite low. So these results actually highlighted the importance of making this process more high throughput, right? So somehow to scale up the processing of individual grains. So we wanted to tackle this problem, and we decided to turn to single-cell sequencing technologies. So these technologies have also grown considerably over the last 10, 20 years, driven by the need from biomedical studies. So basically, these are the kind of technologies that allows us to process genetic material of hundreds to hundreds of thousands of cells in parallel. And it's important to be able to track tissue development, cancer progression, etc. And there's a huge variety of them. So some of them are based on microwells, some of them are based on microfluidics, either microfluidic channels or droplets that we use as reaction chambers. Sometimes even the cell itself can be a reaction chamber in which you're processing DNA. So when we were starting this project a few years ago, we thought, oh, there's going to be a huge number of options to choose from for pollen. But we were very wrong, because there are some specifics of pollen that cells don't have, and that is that they have a tough shell, and so that causes them to sediment faster. They also have all sorts of appendices on those shells, spikes, that makes them clump together and attach to surfaces. And they also come in huge diversity, when single-cell technologies are normally developed for a monoculture, so all the cells are the same in a single sample. So it actually took us years to try different instrumental setups, and we had many setbacks, many failures. But I'm very happy to report that recently we have found an instrumental setup that we believe would be appropriate, not just for pollen, but for other microfossils. So it's basically an instrument called Selector by company MMI, and it's a semi-automated, microscopy-based, a single-cell manipulator. And we are using 2 modules from this instrument. One basically has microcapillaries, so it's kind of following on Parducci's work, but this is a semi-automated way, so it's not the human who has to handle the pipette, it's actually the robotics on the microscope. We just have to pick it on the screen. And then the next step, it also has a UV laser, which can break nicely through pollen, and we also believe, we haven't tried it on other microfossils, but if it goes through pollen, it probably will go through other things as well. So it's early days, we just got this instrument in February, so there's a lot of optimization ahead of us, but we are optimistic. Okay, so I'm just going to finish with some future directions. Apart from the methodological ones that I just talked about, there are some, let's say, conceptual or question-driven directions. The first one is that we are trying to push temporal boundaries of DNA retrieval from sediments. So at the moment, the record breaker is the 2.3 million year old record from Copenhagen in Greenland. But we are trying to go several million years, potentially maybe dozens of millions of years in the past. And the rationale behind these efforts is that if we get genetic material so far into the past, we might find one that changes our understanding of evolutionary or an ecological history. Not every record will do that, but as it did in the case of Mastodon, some will. So that's one important direction. Another one, and I think it might be actually close to heart to paleoecologists here, and that is to reconstruct species interactions. So the proxies we have to date, including environmental DNA, they can give us co-occurrence of species. But just because species co-occur together doesn't mean that they are interacting. And just because we know how they are interacting today doesn't mean that that's the way how they were interacting in the past. So we need an independent way of reconstructing past species interactions. So we have tried to do that, again, in Wang et al. 2021 Nature paper, but it was more just looking at effects of one variable, one type of organism in an ecosystem to another. We are now looking at other ways, other bioinformatic methods that we could use to get a better handle on these interactions. Another way of doing potentially this is, instead of trying to solve it computationally, is to solve it at the level of the wet lab again. So we have a new project funded by Novo Nordisk that is trying to develop a method to measure stable isotope ratios directly on environmental DNA. So the idea is to extract DNA, then separate the DNA that belongs to a taxa of interest, multiple taxas of interest if we're looking at interactions, and then measure carbon, nitrogen, oxygen, and hydrogen, which are all present in DNA molecule, and so get those measures. As I don't know if there are people here using stable isotopes, but stable isotope analysis is one of the most powerful analysis we have at the moment for reconstructing past species interactions, but so far it has only been done on tissues. So now we would want to try to do this on DNA directly. And finally, the third direction that is very important in the center where I'm part of, in Copenhagen, the Center for Ancient Environmental Genomics, is to actually apply ancient environmental DNA to make a societal impact. So we have a new initiative called Ancient Environmental Genomics Initiative for Sustainability, or AEGIS for short. And its aim is to try to actually promote food security in the face of climate change by building more resilient crops and cropping systems. And the way we will try to do this is that we're going to draw on the huge variation, genetic variation that we have from the past, from past crops, cropping systems, and wild ecosystems, find those genetic variants and functional traits that are associated with favorable responses of species and ecosystems, and try to incorporate that into present-day crops and cropping systems. Okay, just want to acknowledge all the wonderful people I worked with over the years, and thank you for your attention. I'll stop here.
Are there any questions?
Great talk, Anna. What's the reason for such a poor success rate in amplifying the DNA from the single grain pollen? Is it preservation, or is it just that like because they're old, or is it something else?
Yeah. The short answer is we don't know really at the moment. We're looking into it. Logically, it's probably because we're starting with very small amount of DNA. In pollen, you have 2 to maybe a dozen of cells, while, from a gram of sediment, that's a much larger sample. So I think that's the starting challenge. So you have to be much more efficient with how you retrieve the DNA and preserve it and take it through the protocol, right? So the other potential reason is also that particular study, they used PCR amplification, rather than building genomic libraries, which we now know is more efficient at capturing small amounts of DNA. So this is something we're working on at the moment, and we will see whether that also changes the success rate. The other thing is that sometimes, maybe on the microscope a grain looks good, you don't see the cracks, but it could be the DNA already left the grain, and so there's nothing really to retrieve there. So
thank you for your review and about the quality and the power of eDNA in paleo environmental studies. I have a question. I mean, in traditional, I would say, paleo studies, we use biomarkers, other traces based on geochemistry, for example, or even microfossils are very much used to reconstruct paleo conditions. And at the same time, we are using the concentration. So we are trying to relate the quantity of biomarker or the abundance of pollen to the conditions and the abundance of the vegetation. In the case of eDNA, for the moment, you are stuck with presence or absence. I mean, knowing that there was one mastodon that made it to Greenland is exceptional. It's very impressive, but at the same time, how many were there during that time? So is there any hope that you would be one day a bit more quantitative, I mean, trying to convert the presence, absence, and the DNA in terms of fluxes or concentration or abundance I mean, it was, for example, you showed the example of the sea ice and the comparison with biomarkers that are used for sea ice as a sea ice proxy. This is exactly the type of work that would be complementary.
yes, thank you. It's a good question. There's actually two ways of processing DNA after the extraction. We can do meta-barcoding, or we can do the metagenomics approach. With barcoding, that's where actually we don't get any measure of abundance. But when we're doing metagenomics afterwards, because we capture all the reads, so we can normalize the number of reads through the record, and we can get some idea of the relative changes in the number of reads belonging, for example, to mammoth or some other taxa. It has its own, I'm sure, taphonomic bias that we don't understand very well at the moment. We're working on it the same way as this work had to be done for pollen, because there's, of course, bias there too, and with other proxies. But that's one way, maybe a more kind of, maybe less robust. The other way is on the bioinformatics side. Actually, by looking at genetic changes through time, we can get effective population size. That wouldn't be a total size, but it will be the size of the reproducing population. And that's something that if we have enough reads, and if they are hitting the right parts of the genome, theoretically should be possible to do, but for that we need to develop specific methods and tools in computational analysis to do that. So we're trying.
Thanks a lot for this very nice talk. My question is about the preservation of DNA, because there have been many attempts where DNA couldn't be measured. So I don't know if it's because of the technique, which wasn't good enough at this time. But do we know about the range of parameters, such as temperature or the content of water, that might destroy the DNA, actually?
I think we have kind of a more general idea rather than a quantitative estimate. So we know that the colder it is, the drier it is, the better it's for DNA. Salinity, high salinity is also good for DNA preservation. But we're actually still in the phase of explorative phase, when we're learning about where we're finding DNA and then kind of backtracking, okay, what are the conditions? Why are we finding DNA here? Or we're retrieving it, and in other places we don't. So we're still in that kind of learning process. But yes, ultimately that's exactly what we would want. We would want to be able to say, okay, in this place, because we know we had these conditions in the past, we have this chance of finding DNA. The thing is, at the moment, we have a pipeline that can process at the University of Copenhagen about 1,000 samples per week. So in a lot of ways, if the cost is not a problem, it's faster to actually just try a sample rather than saying, oh, based on everything we know so far, this is a low chance, so we won't even try it, especially if it's a precious sample. So that's the kind of a strategy we have at the moment. Maybe not all groups, but that's what we do. But ultimately, yes, we want to understand better why some DNA in some places gets preserved better than not. Yeah.
Hi, thanks a lot for a great talk. My question was about what you were saying about pushing the boundaries back in time to millions of years ago. How do you deal with the fact that lots of the DNA fragments that you're going to find millions of years ago may not have a modern genome available? Like, how do you compare the ancient DNA fragments with your modern database?
Yes, no, that's a very good point. So yes, once we go beyond 1 or 2 million, we could be dealing with species that went extinct, or basically populations that are very different from today. That's why I think as we go back in time, the kind of taxonomic level to which we can assign the reads also kind of increases. So we go from species to genus to potentially family. We are, in case of course, mammoth, because we have the remains, we can actually get the DNA from the remains, create a reference genome of mammoth, and then trace it back. But of course, for example, many plant species, we can't do that because we don't have them. So yeah, that's definitely something that limits what we can do at the moment. Maybe there is some solution on the horizon, but not at the moment.
Thanks for your very nice talk. My question is very similar to Anne-Alexandre, but more on the extraction side of things. I was wondering, do you know your limitations in extracting DNA from different samples? Like, are some species or organisms less easy to extract, or are some DNAs more adsorbable than others?
Yeah. So actually, Karina's work that I mentioned earlier on DNA mineral interactions, what she found is that some minerals are better at absorbing DNA than others, which on one side is good because it means that when DNA gets absorbed to mineral, it gets protected from the enzymes of microbial communities around it and from hydrolytic attacks from water and from sunlight and other things. So that's a good thing. But then actually the sort of the typical extraction protocols that we started from the modern genetics, they're not as effective to get it from these minerals, right? Because they tie DNA so strongly that the standard buffers don't actually detach DNA. And this is what we realized, that, for example, clay sediments, that we might be, it's hard to say what percentage of DNA we're getting, and the rest just stays on the clay and we lose it. So her group is developing new protocols with new buffers that will allow us to actually get more. So even from these minerals that tightly bind DNA, how to actually release, break those relationships and release DNA. Yeah, so that's also work in progress.
Thank you for your talk. We know that if you take fresh genetic material, and as you point out, it sorbs to clays or something that protects it. So you can take fresh genetic material, it can move on a clay, and let's say a bacterial bit of DNA or an RNA with microbial resistance. And we know that microbial resistance, that can be expressed somewhere else when that particle arrives there and it offloads. So my question is with this very ancient genetic material, is it accessible to current organisms, and if not, why not?
It's a very good question. This is actually exactly what Karina is looking at. One of the questions she's trying to answer is how actually antibiotic resistance is tying to this mineral DNA interactions. And of course, if you have a horizontal gene transfer and the bacteria can access DNA attached to minerals, then they could actually get those genes, that potentially very old genes that were lost from a population, genes that we don't want to go back to bacterial populations. So her results suggests that, yeah, that's happening. At what rate, and what is really affecting it, it's still very much, she has actually more or less started this kind of line of questioning. So I wouldn't be able to tell you exactly, there's no answer at the moment, but we're definitely seeing that it is possible to have horizontal gene transfer, through minerals.
Thank you for a great talk. You've shown us how difficult it is to get DNA from pollens, and you say that it might not be the case for other types of microfossils. But I was wondering if, when it comes to reconstructing a full ecosystem from sedimentary DNA, if you've already identified blind spots, and by this I mean types of organism or species that we already know that it's going to be very difficult to get DNA from.
Yes, so we have some idea already. So what we find in ancient environmental sample is very much shaped by the biomass. So the larger the biomass the species has, the more DNA it's going to contribute, right, to a lake sediment. So we find generally more, of course, microbial DNA dominates. After that, it's more often plants. So we have quite a bit of plant DNA. Animals are kind of many animal groups are the blind spot. So we get very few birds, we get few insects, we get actually mammals, probably more than birds and insects. So so there is definitely groups that we are not getting, and that's just probably because they don't have the biomass to make the impact, to make a significant contribution to the ancient DNA pool, or because the way that their DNA is coming into the lake is also not at the same amount. Because, for example, from plants, we can see that by erosion, a lot of that material come in. With animal DNA, probably less so. So that's also maybe a factor. But it's a very good question. We are still looking into it.
Okay, thank you.